Hello students, this is your science teacher Aparna. We are studying chapter number 9, reproduction in animals. So in this chapter, till now we have studied about sexual reproduction, because when I started the chapter, then I told you about what is reproduction, what are the modes of reproduction. Then there were two modes of reproduction, asexual reproduction and sexual reproduction. Then firstly we have focused in sexual reproduction and how it is going on, and what is the result of fertilization, and how zygote is formed, then how it is developed as embryo, and how it developed as fetus, and how become it a baby. Okay, and now today we will focus on asexual reproduction. So when I started that chapter, then I told you that only that is a difference between asexual reproduction and sexual reproduction, that in sexual reproduction two parents are involved, one is female and one is male in sexual reproduction. But when we are talking about asexual reproduction, so there is involvement of only one parent, single parent involvement in asexual reproduction. So you can open the page number 106 in your book. So asexual reproduction 9.3. So far we have learned about reproduction in some familiar animals. But what about very small animals like hydra and microscopic organisms like amoeba? So amoeba is a microscopic organism and hydra is also very small in size. So what is going on in these animals? So you can see the picture of hydra. So here what is going on here? There is a bud that is known as bud. Okay, that is known as bud. And after that, in each step, that bud is growing. Okay, and at the last, this bud is producing a new one. Okay, this bud is producing a new one, and this production is going on by a bud. That's why it is known as budding. And in hydra, what is the mode of reproduction? Budding in hydra. You can consider this picture as budding in hydra. So in hydra, what is going on? A bud is there, and after that it is converting into a new hydra. So this process is known as budding. And what is the mode of reproduction in hydra? Budding process. Now come to the amoeba. Amoeba is also microscopic animal. It is unicellular, you know. So and you also know that is irregular in shape. So you can see the picture of amoeba. So this is the parent amoeba. Okay, you can consider it as parent amoeba. And what is going on? What is going on here? Now division of nucleus is started, and how it is started to shrink from the center. Okay, from the center it is started to shrink, and structure is just like that, and nucleus is about to divide. Okay, and in next step you can see that nucleus is already divided. Nucleus is already divided, and after that it will be break from center. So what is going on here? Fission. Fission, that means breaking into that means breaking, and binary fission, that means breaking into two. Okay, breaking into binary fission means breaking into two. So you can say now these are the daughter amoeba, and only single parent involved here. So that is also mode of an example of asexual reproduction. So in amoeba, what is the mode of reproduction? Binary fission. That means a single amoeba is breaking into two daughter amoeba cells. So here in hydra only one parent is involved. In amoeba only one parent is involved. That means these are the example of asexual reproduction, and here reproduction is going on by bud, so that's why it is known as budding. And here a single cell is dividing into two daughter cells, that's why it is known as binary fission. So these were the example of asexual reproduction. So by these two topics this chapter is completed, and at the last there is a story given about Dolly, that was a clone, and that was a first clone which was done by Ian Gilbert and his colleagues, and where It was done in a lab of Scotland. Then what they have done, they have taken two species of sheep and they have interchanged the nucleus of egg. And when what they have done, they have changed the egg of blackfish sheep. Okay, they have changed the nucleus of egg of blackfish sheep, and from where they have taken another nucleus from the whitefish sheep, and the name of the sheep was, just a minute, yes. During the process of cloning Dolly, a cell was collected from the mammary gland of a female fin dorsate sheep. So one species name was fin dorsate sheep, and egg was obtained from Scottish blackfish sheep. Okay, and the nucleus was removed from the egg. Then the nucleus of a mammary gland cell from the fin dorsate sheep was inserted into the egg of the Scottish blackfish sheep, whose nucleus had been removed. So here what was going on? Change of nucleus. The egg that produced was implanted into the Scottish blackfish ewe. Development of this egg followed normally, and finally Dolly was born. Though Dolly was given birth by the Scottish blackfish ewe, it was found to be absolutely identical to the fin dorsate sheep. So what was going on here? That means who has given birth? Blackfish sheep. Okay, but she was identical like fin dorsate sheep. Okay, fin dorsate sheep, she was identical because nucleus was inserted of fin dorsate sheep. So that was the cloning. Actually, what is cloning? Just like photostat, if you want to photocopy anything, so exactly same copy. Okay, cloning means exactly same copy. So Dolly was exactly identical like fin dorsate sheep. However, it is given birth by blackfish sheep. Okay, so that is cloning. So Dolly was the first clone, first mammal clone. Okay, and when Dolly was born on 5th July 1996, and it was the first mammal to be cloned. Dolly was a healthy clone of the fin dorsate sheep and produced several offspring of her own through normal sexual means. Unfortunately, Dolly died on 14th Feb 2003 due to a certain lung disease. Since Dolly, several attempts have been made to produce cloned mammals. However, many die before birth or die soon after birth. The cloned animals are many a times found to be born with severe abnormalities. Okay, so I hope you have understand the chapter. Now this chapter is completed, and from tomorrow we will start the writing work. Till then, watch and listen it carefully. If you have any type of doubt, please ask. Thank you. Have a nice day.